You begin by settling into stillness, as if your thoughts are snowflakes gently lowering themselves onto a quiet winter field. Your breathing slows. Your attention turns inward, not toward emotion or memory, but toward something smaller, far smaller, and yet more complete than any story you have ever known. Inside you, there is a library. Not made of wood or stone, not lit by lamps or windows, but by chemistry and motion and patient repetition. It exists in every living cell of your body, trillions of rooms, each holding a similar collection. And in each room rests a coiled manuscript so long and detailed that if you stretched it out, it would measure about 2 meters from end to end, yet it fits inside a space too small to see. You walk there now in your imagination, quietly, respectfully. This is your genetic library, your DNA. It does not use ink. It does not use paper. It uses molecules shaped like curved ladders, gently twisted, like a spiral staircase turning upward forever. Scientists call this shape the double helix, but tonight you can simply think of it as a winding staircase of instructions, steps made of paired letters, rising and turning in a soft, endless rhythm. You do not hear noise here. No echoes. No footsteps. Only the slow turning of processes that never sleep. Every shelf holds a sequence. Every sequence holds a pattern. And every pattern is written in just 4 chemical letters. Only 4. Imagine every poem, every manual, every map, every recipe you have ever seen written using only 4 symbols arranged in different orders. Simple pieces, infinite meanings. These 4 letters are not A, B, C, D, but close cousins, A, T, C, and G. They stand for molecules with longer names, but you do not need those names tonight. What matters is their partnership. A always pairs with T, C always pairs with G. Like dancers who never change partners, they meet across the spiral steps and hold the structure steady. You drift past these shelves and notice something comforting. Nothing here is rushed. Your body is not hurried at this level. Cells read instructions slowly. They copy with care. They pause and check their work. Life, at its deepest layer, is not frantic. It is methodical and gentle. Each time a cell prepares to divide, it opens the staircase down the middle, like a zipper being drawn apart with delicate fingers. Each side becomes a template, a guide for building a new matching half. New letters float in, attach to their partners, and rebuild what was opened. 2 staircases now stand where one stood before. Copying is constant. Copying is calm. Copying is sacred work. You watch as molecular helpers, enzymes, move along the strands. You can picture them as careful librarians wearing soft gloves, sliding along the text, making sure each new letter matches its partner. They do not understand meaning the way you understand meaning, but they understand fit, shape, complement, belonging. This is how your cells remember you. Not your memories, but your structure, your eye color, your enzyme shapes, your cellular tools, the way your hemoglobin holds oxygen, the way your skin rebuilds, the way your neurons grow their branching trees. Identity here is not stored as photographs or sentences, but as patterns of assembly. You are written as instructions for building yourself. And like any library, this one is used often, frequently opened, frequently copied. pages handled again and again. And whenever information is copied even carefully, there is the possibility of a tiny change. Not damage. Not disaster. Just change. You might imagine a monk copying a manuscript by candlelight centuries ago. Most letters are reproduced perfectly. But once in a long while, a stroke is slightly darker, a curve slightly shorter, a dot placed a breath too far to the left. The sentence still stands. The meaning often survives. But the copy is not perfectly identical. In your cells, something similar can happen. When DNA is copied, the enzymes are astonishingly accurate, so accurate that errors are rare. Extremely rare. But rare is not the same as never. Out of billions of letters, a few may shift. One letter may be replaced with another. One may be skipped. One may be added. A tiny edit in a vast book. You pause in the aisle and feel the scale of this. Your entire body built from text. Your living form arising from molecular spelling. It feels both intimate and immense, like discovering that every wave in the ocean is written by invisible poetry beneath the surface. Most edits do nothing noticeable. Many occur in stretches of DNA that do not directly build proteins. Some changes still produce the same result, because the code has built-in redundancy, like synonyms in a language. Different spellings, same meaning. This too is comforting. Biology expects variation. It plans for it. You continue walking and notice that this library is not static. It is alive with readers. Molecular machines arrive, copy a section, and carry the message outward to the cell's factories, where proteins are assembled like folded origami tools. DNA stays safely in the archive. Working copies travel outward. The original text is preserved whenever possible. There is wisdom in that design: protect the master copy, send out the duplicates. And yet, even master copies are not locked away forever. They must be opened. They must be read. They must be duplicated when cells divide. Life requires access. Access invites possibility. Possibility allows change. You run your fingers in imagination along the spiral staircase rail and realize something gentle and profound. Perfection is not the requirement for life to continue. Function is. Continuity is. Adaptation is. The library does not demand flawless duplication. It allows small differences. It tolerates tiny surprises. Sometimes it even benefits from them. A single letter change can be silent, like replacing color with color. Nothing truly lost. Sometimes it shifts emphasis, like changing warm to hot. Noticeable, but manageable. And sometimes, more rarely, it changes the whole instruction, altering the shape of what gets built. But you are not there yet in the story. For now, you simply rest inside the awareness that within you lives a patient archive, written in chemistry, copied in calmness, and guarded by layers of care. A living manuscript, continuously read. And somewhere, softly, invisibly, a single letter waits to be noticed. You return to the inner library, but this time you move closer to the text itself. Not the shelves. Not the spiral staircase as a whole, but the letters. The smallest readable pieces. The alphabet of your living design. Imagine leaning close enough to see that the entire manuscript of your body is written with only 4 symbols. Just 4 repeating characters arranged in long, winding sequences. It feels impossible at first. How can something as complex as you arise from such a small alphabet? And yet complexity often grows from simple rules repeated with patience. You already met their initials, A, T, C, and G. Tonight, you sit with them like quiet companions. Each letter is a molecule, a shape with edges and curves, tiny hooks and pockets where another shape can rest. They are not chosen by sound or meaning, but by fit. Like puzzle pieces that recognize their partners by touch alone. A always pairs with T, C always pairs with G. No debate. No hesitation. Attraction by structure. You can picture them as dancers in a slow ballroom, always finding the same partner across the floor. When A extends a hand, only T can take it comfortably. When C turns, only G matches the movement. The dance repeats billions upon billions of times inside you, steady and dependable. This pairing is called complementarity, but you don't need the word to feel the idea. It is the comfort of things that belong together. As you observe, you notice that each pair forms one step in the spiral staircase of DNA. A with T makes a step. C with G makes a step. Step after step after step, the staircase rises, twisting gently, storing instructions in its order. Not in the individual letters, but in their sequence. Sequence is meaning here. Like beads on a string, the order creates the message. Change the order, and you change the instruction. Keep the order, and the meaning holds steady even across generations. You drift alongside a stretch of this molecular text and imagine it like music written on a staff. For notes only, yet endless songs. Some sequences signal where a gene begins. Some signal where it ends. Some describe how to build a protein, one molecular building block at a time. Proteins, those folded, working shapes, are the tools and structures of your body. Enzymes, receptors, fibers, messengers. The DNA alphabet does not build them directly. Instead, it writes recipes for assembling them. You watch as a small molecular reader attaches to the DNA strand. It does not read the whole library, only a selected passage. It moves along slowly, like a fingertip tracing braille, recognizing patterns without awareness, without thought, yet with perfect consistency. This reader builds a working copy, a temporary ribbon called RNA. If DNA is the archive book, RNA is the notepad copy carried into the workshop. The original stays protected. The copy travels outward. There is something deeply soothing in this layered design. Preserve the master, use the duplicate. Nature builds with backups. The RNA copy uses a nearly identical alphabet, with one small variation, one letter swapped for a close cousin. Not a disruption, just a dialect change. The message remains readable. The work continues. You notice that the letters are read not one by one, but in groups of 3. 3-letter words. Triplets. Each triplet signals one building block of a protein and amino acid. It is like reading instructions in 3-letter beats, steady and rhythmic. 3 letters. 1 unit. Repeat. You begin to sense the pulse of this system, triplet by triplet, addition by addition, like footsteps on a quiet path. No rushing, no leaps, just ordered progression. And here is another gentle wonder: multiple different triplets can sometimes signal the same amino acid. The code contains synonyms. Built-in tolerance. If one letter changes but the triplet still points to the same building block, nothing changes in the final structure. It is like spelling color or color. Different letters, same meaning. Redundancy is not inefficiency here, it is resilience. A cushion against small errors. A soft landing built into the language of life. You rest with that idea for a moment. Your biology expects variation and prepares for it in advance. The alphabet is strict in pairing, but flexible in expression. You float further and notice stretches of DNA that do not code for proteins at all. Once, scientists thought these regions were useless, silent filler between meaningful lines. But over time, understanding softened. Many of these regions regulate timing, volume, and coordination. They are like punctuation, spacing, and tempo marks in music. Not every symbol builds a structure. Some shape the performance. The deeper you drift, the more the alphabet feels less like machinery and more like poetry written in molecules, patterned, constrained, expressive through order rather than vocabulary size. You begin to imagine how copying happens again: the strand opening, letters exposed, partners arriving from the surrounding fluid. Each free-floating letter does not guess where to attach. Chemistry guides it. Only the correct partner fits cleanly. Attraction and geometry decide. This is recognition without awareness. Accuracy without intention. And yet, you remember, not every pairing is perfect every time. The system is highly accurate, but reality is physical, not magical. Molecules move. Temperatures shift. Energies fluctuate. Occasionally, a letter that almost fits may briefly attach. Usually, it is caught and corrected. There are molecular proofreaders, you will meet them more closely later, who scan and repair mismatches. But sometimes, before correction arrives, the strand closes and the near fit becomes permanent. A tiny mispair. You do not feel alarm here. Only curiosity. Because most mispairs occur in places where they change nothing noticeable. Some occur where redundancy protects the meaning. And some will be repaired later anyway by dedicated repair systems. Still, the possibility exists. A whisper of variation at the alphabet level. You sit beside a single step of the spiral staircase, one paired set of letters, and imagine how small it truly is. Swap it, and the structure still stands. The staircase still turns. The library still holds. And yet, downstream, far along the reading path, that tiny change might alter a word, which might alter a building step, which might slightly reshape a folded protein. Small origin. Distant effect. Like changing one note in a long symphony, perhaps unnoticed, perhaps transformative. Your breathing stays slow as you consider this. Life is written in an alphabet of 4. Stability comes from pairing. Meaning comes from order. Resilience comes from redundancy. And change, when it happens, begins quietly at the level of a single letter meeting the wrong partner for just a moment. You close your eyes in this inner library and listen not to sound, but to pattern, and you begin to wonder, gently, what happens during the long night of copying, when the readers move, the letters pair, and somewhere in the dim molecular candlelight, one character chooses differently than expected. You drift back into the inner archive, but now it is evening there, not darkness, not sleep, but a warm, amber stillness, like a hall lit by candles and patient hands. This is the hour of copying. The hour when the manuscript of you is prepared to be shared. A cell is getting ready to divide. Division is not violence here. It is inheritance. One room becoming two rooms, each needing a complete library of instructions. Nothing can be left behind. Every letter must be available in both places. So the great spiral staircase must be copied from end to end. You stand beside it like a quiet observer as the work begins. The double strand does not snap apart, it opens gradually, like a zipper drawn with reverence. A molecular guide moves to the starting point, a special sequence that signals, begin here. You can imagine it as a golden ribbon tied around a page corner. Start markers exist throughout your DNA, placed with exquisite specificity. An enzyme arrives, helicase, though tonight you might think of it simply as the opener. It glides forward, gently separating the paired letters, breaking the soft chemical handshakes between partners. Not destroying, just parting. The staircase becomes two rails, each exposed, each ready to guide a new partner line. The opening creates a shape like a fork in a path. Scientists call it a replication fork, but you can see it as a candlelit writing desk where two scribes work outward in opposite directions. Nothing rushes. Accuracy matters more than speed. New letters float nearby in the cellular fluid, like loose type pieces in a printer's tray. They do not attach randomly. They wait for their matching partners to be revealed along the open strand. When an A appears, AT is drawn close. When AC appears, AG settles in. Attraction by fit. Selection by shape. Another enzyme steps in, DNA polymerase, the master scribe. It does not think, yet it is astonishingly selective. It slides along the exposed strand and adds each new letter to the growing copy, one at a time, always checking the match before sealing it into place. You watch the motion, steady, deliberate, almost meditative. Add, check, seal. Add, check, seal. It feels like watching a calligrapher paint characters with a fine brush. No wasted strokes. No flourish. Just consistency. But copying is not as simple as tracing a line. The two original strands run in opposite directions, like two roads with opposite traffic flow. This creates a small complication in the choreography. One side can be copied smoothly, continuously, following the opening fork. The other must be copied in short segments, working backward in small patches. You see the second scribe working in fragments, write a short stretch, pause, reposition, write another. These fragments are later joined together into a continuous strand, stitched with molecular glue. Even in biology, some work is done in pieces before it becomes whole. There is no frustration in this, only adaptation. You notice helpers gathering, stabilizing proteins that hold the open strands apart so they do not snap back together too soon. Clamp proteins that keep the polymerase attached so it does not drift away. Coordinators that manage timing. It is not a single worker but a quiet team, each with one role, each performing without ego. Replication is collaboration. And through it all, proofreading is constant. The polymerase does not only write, it also reviews. Each time it adds a letter, it senses whether the fit is truly correct. If the geometry feels wrong, too loose, too strained, it pauses. The letter is removed. A better match is tried. This built-in proofreading makes copying extraordinarily accurate, far more accurate than most human copying could ever be. You might imagine the scribe leaning back after each word, squinting gently, and adjusting his stroke before the ink dries. Because of this, the error rate is incredibly low. Out of billions of letters, only a few may slip past unnoticed. The system is not careless. It is devoted. Still, this is a physical universe. Molecules vibrate. Energies fluctuate. Occasionally, a near match slides in just long enough to avoid immediate detection. The shape is close enough. The seal is made. The line continues. The candle flickers. The page turns. You feel no alarm, only awe at the balance. Absolute perfection would require infinite time and energy. Life chooses remarkable accuracy instead, accuracy sufficient for continuity, flexible enough for change. As copying continues, you notice timing signals embedded in the DNA sequences that say slow down, pause, stop here. The manuscript contains instructions not only for building proteins, but for managing its own duplication. The text helps guide the copying of the text. Self-referential. Self-regulating. Quietly brilliant. Hours pass in cellular time, though to you it feels like watching snowfall. Eventually, the entire length is copied. 2 complete double strands now rest where one once lay. Sister manuscripts. Nearly identical. Nearly. Before the process is declared complete, additional inspectors arrive, repair enzymes that scan the new strands for distortions. They patrol like nighttime caretakers walking library aisles. If a mismatch bulges the shape of the spiral even slightly, they detect it. The incorrect letter is clipped out. The gap is filled correctly. The backbone is resealed. You feel wrapped in reassurance. There are layers of checking, layers of care. Your biology assumes responsibility for accuracy again and again. And yet, despite proofreading during writing and repair after writing, a tiny number of changes remain. Not many. Sometimes just one in millions of letters. A statistical whisper. A molecular footnote. Most will never matter. Some occur in regions that are never read into proteins. Some produce synonymous triplets, different spelling, same instruction. Silence an outcome. But once in a long while, a change sits inside an important word. A triplet shifts meaning. A building block in a protein recipe is swapped. The final folded shape adjusts, perhaps subtly, perhaps significantly. All from a moment during copying. You sit with the humility of that. Not fragility, but sensitivity. Life is robust, yet responsive. Structured, yet alive to variation. The candles in the copying hall burn low now. The strands rewind into their protected coils. The enzymes disperse. the cell prepares for division, carrying forward its paired libraries into 2 daughter rooms. Inheritance complete. But you remain seated a moment longer beside the manuscript, sensing that copying is not merely duplication, it is also the birthplace of difference. Every generation of cells carries continuity and the faint possibility of novelty born in these quiet acts of molecular transcription. And somewhere, in the glow of careful replication, a single letter written just slightly differently waits to reveal what even the most careful copying can become. You remain in the library, but now your attention grows more precise, like leaning closer to a single line of text instead of the whole page. The copying is finished. The candles are low. The new manuscript rests beside the old, almost indistinguishable. Almost. Tonight, you are here to notice the smallest kind of change. Not a torn page. Not a missing chapter. Just one letter, quietly different from what came before. You might imagine a long sentence written across a scroll. Thousands upon thousands of characters flowing without spaces. Now imagine that somewhere in the middle, one character is replaced. Not erased, simply swapped. The sentence still exists. The scroll is still readable. But something subtle or significant has shifted. In genetics, this is often called a point mutation. A single letter substitution in the great molecular text. One base replaced by another. A where a G once stood. A C where a T used to rest. You sit with how small that really is. One step on a staircase of billions. One bead changed in a necklace the length of a continent. And yet meaning in biology, like meaning in language, depends on order. You remember that the code is read in triplets, 3-letter words that specify which amino acid is added when building a protein. These triplets are called codons, but tonight you can think of them as 3-note chords. Change one note, and sometimes the chord still sounds the same. Sometimes it becomes a related harmony. And sometimes it becomes a completely different sound. You watch an example form in your mind. A 3-letter genetic word calls for a particular amino acid, one specific building block in a growing protein chain. If a single letter in that word changes, 3 different outcomes are possible. The first is the gentlest, nothing changes in the final protein at all. This is called a silent change, and the name is kind. Because of redundancy in the genetic code, multiple codons can point to the same amino acid. Different spellings, same ingredient. Like writing theater or theater. The reader understands either way. The protein is built exactly as before. A silent mutation is like a whisper absorbed by a thick curtain. It happens, but it does not travel far. The second outcome is a swap, one amino acid replaced by another. The sentence changes a word, but not its grammar. The protein is built with one different piece in one position. Sometimes the difference barely matters, especially if the replacement amino acid has similar properties. Similar size. Similar charge. Similar behavior. It is like replacing a wooden chair with another wooden chair of slightly different design. The room still functions. But sometimes the replacement is more dramatic, like swapping a wooden chair for a tall metal stool. The structure still stands, but comfort, balance, or interaction may change. Proteins are folded shapes, and their function depends on that folding. One different building block can subtly alter how the chain bends, twists, or locks into place. The third outcome is the most disruptive, a change that turns a meaningful codon into a stop signal. You remember that proteins are built like sentences with punctuation. There are codons that mean start here and codons that mean stop here. If a single letter substitution converts a building instruction into a stop instruction, the protein is cut short, unfinished. A sentence ending mid-thought. Sometimes a shortened protein cannot function at all. Sometimes it functions weakly. Sometimes the cell notices and recycles it. There is sadness here, but not drama, only consequence. You breathe slowly and notice how biology is not offended by change. It simply responds. You walk further along the strand and observe that not all DNA regions are equally sensitive. Some sequences are crucial, deeply conserved across species and time. Changes there are more likely to have noticeable effects. Other regions are more flexible, like margins with room for variation. Context matters. Position matters. Neighborhood matters. A letter change in a critical active site, the working center of an enzyme, can alter how it binds to its target. A letter change in a regulatory region can alter how much of a protein is produced, turning volume up or down. A letter change in a rarely used stretch may pass through generations unnoticed. The same size change, different outcomes depending on location. You sit with this like a meditation on sensitivity. In your own life, a small change in the right place can redirect a path. A single conversation. A single decision. Scale is not everything. Placement is. Cells understand this quietly. They do not panic at substitution. They evaluate results. You watch a protein being built from a slightly altered recipe. The chain emerges from the ribosome, the cell's protein workshop, like thread from a loom. It begins folding immediately, guided by chemistry and environment. Folding is not random. It is driven by attraction and repulsion, by water-fearing surfaces tucking inward and water-loving surfaces facing outward. Shape becomes function. If the substituted amino acid sits on the surface far from a working site, the fold may remain nearly identical. If it sits at a hinge point or binding pocket, the motion may change. Grip may loosen. Timing may shift. This is not failure. It is variation expressed as physics. You notice too that cells often make many copies of the same protein. Populations, not singletons. If one copy functions less efficiently, others may compensate. Biology builds with redundancy again and again, backup upon backup, like multiple lanterns lighting the same path. You feel the kindness in that design. And yet, occasionally, a single letter change produces a protein with a new behavior, not worse, not broken, simply different. Different stability. Different activity level. Different interaction. Most differences are neutral in the grand balance. Some are harmful. A few are beneficial in certain environments. Outcome depends on surroundings as much as structure. You rest beside the altered letter now, just one changed run in the spiral, and sense how quiet the event truly is. No alarm bells. No flashing signals. Just a different partner chosen during copying, sealed into place, carried forward. From here, consequences unfold slowly, through use in context and time. You realize something soft and steady. Mutation is not an intrusion from outside life, it is part of how life explores possibility. Not intention, but allowance. Not chaos, but gentle experimentation within strong boundaries. The library does not crumble when a letter changes. It adapts its reading. And as you close your eyes in this molecular hall, you begin to wonder how many kinds of small edits exist beyond simple substitution, not just changed letters, but missing ones, added ones, shifted rhythms, like different ways a poem can be revised without rewriting the whole page. You remain seated in the quiet archive, but now the view widens. Until now, you have been watching a single letter change, one substitution, one exchanged character in a vast molecular sentence. Tonight, you discover that change has more than one face. Mutation is not a single event, but a small family of possibilities, different kinds of edits made in the text of life. You can think of DNA as a long poem written without spaces. Continuous. Flowing. Measured in triplets. And like any poem copied across generations, edits can appear in different forms, not only replaced letters, but missing ones, extra ones, shifted ones. You walk slowly past 3 doorways, each opening onto a different style of change. The first doorway is the one you already know, substitution. One letter replaced by another. A swap. The rhythm of the sentence stays the same length. Only a single character differs. Often gentle. Often quiet. Sometimes meaningful. You have already felt how a substitution can be silent, or expressive, or disruptive depending on where it lands. You nod to it like an old acquaintance and move on. At the second doorway, something slightly more noticeable happens. A letter is missing. This is called a deletion, but tonight you can imagine it as a skipped syllable in a spoken line. The reader moves forward, but one sound is gone. If the deletion happens in a stretch of DNA that is not used to build proteins, the effect may be minimal or none at all. But if it happens inside a coding region, inside the triplet rhythm, the consequences can ripple. Because the code is read in groups of 3, removing one letter shifts the reading frame. Every triplet downstream is regrouped differently. The words change not just at the site of deletion, but everywhere after it. You picture a sentence like, "The cat ate the pie." Remove the C from cat, and the grouping becomes, "The ate hap ie." The letters are mostly the same, but the meaning dissolves. This is called a frame shift, and it is one of the more powerful ways a small edit can reshape a message. Not louder, just more pervasive. You breathe slowly and remember, powerful does not always mean catastrophic. Often, a frameshift protein is simply recognized as non-functional and quietly recycled by the cell. Cleared away. No drama. Just housekeeping. Cells are practical readers. At the third doorway, you see the opposite of deletion. Instead of a missing letter, there is an extra one, an insertion. A new character added where none existed before. Like an extra beat placed into a line of music. Again, if the insertion happens outside protein coding regions, it may pass like a breeze through tall grass movement without consequence. But if it happens within a coding sequence, and if the number of inserted letters is not a multiple of 3, the reading frame shifts again, just like a deletion. Downstream words reorganize. New triplets form. New amino acids are chosen. The protein recipe changes from that point forward. Deletion and insertion are like removing or adding a step on a spiral staircase. The structure still turns, but your footing changes with every step after. You sit with this image and feel how sensitive pattern systems are to rhythm. Not fragile, but responsive. There are also insertions and deletions that occur in groups of 3 letters, whole codons added or removed. These do not shift the reading frame. Instead, they add or subtract a single amino acid from the protein chain. Like adding or removing one bead from a bracelet. The overall design remains readable, though the final shape may adjust slightly. Not every edit rewrites the paragraph. Some only adjust a phrase. You continue walking and discover another, quieter category: repeat expansions. In some regions of DNA, short sequences are meant to repeat a certain number of times. Like a decorative pattern along a border. During copying, the cellular machinery can sometimes slip and copy the repeat too many times. The pattern grows longer. Repeat expansions are like echoes that become extended. Instead of tap tap tap, the rhythm becomes tap 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 tap. Sometimes this has little effect. Sometimes it changes how the DNA region behaves, how it folds, how it is read, how it interacts with regulatory proteins. You notice that many mutation types are not about destruction, they are about variation in length, rhythm, grouping. Editing styles, not explosions. There are also larger scale changes, whole sections duplicated, inverted, or moved, but you only glimpse these from a distance tonight, like mountain ranges on a horizon. You stay with the smaller edits, the ones closest to the letter level, where the poetry of change feels most intimate. As you wander, you realize something soothing: most mutations are neutral. Not harmful. Not helpful. Simply present. The genome is vast, and much of it can tolerate small variation without altering cellular behavior in any noticeable way. Life continues smoothly. Proteins fold. Signals travel. Energy flows. Biology is buffered. Cells also have layers of response. If a mutation creates a protein that misfolds, quality control systems often recognize the shape as unstable and guide it toward recycling. If a regulatory signal becomes too strong or too weak, feedback networks may soften the effect. Pathways adjust output like dimmer switches, not just on-off buttons. You feel surrounded by cushions, mechanisms that absorb shock and preserve balance. And yet, you do not dismiss mutation as unimportant. Because once in a while, an edit changes the melody. A substitution alters an active site. A frameshift reshapes a receptor. A repeat expansion changes gene regulation. A protein behaves differently enough to influence the cell's choices. Small edits, large echoes. You notice too that mutation does not mean intention. There is no goal in the change itself. It is not trying to improve or disrupt. It is simply variation arising from physical process, copying in a warm, dynamic environment with moving molecules and finite time. Meaning comes later, through interaction and consequence. This feels gentle, change without motive. You sit again beside the long manuscript and imagine it not as rigid law, but as living literature copied across billions of cellular generations, occasionally edited, usually preserved, always used. Stability is the rule. Variation is the whisper at the edge of the page. And as your breathing slows, you sense that if edits EXIU remain inside the living library, but the hour has shifted. Copying is done. The scribes have rolled up their sleeves and stepped away. Now comes the quieter shift, the caretakers, the inspectors, the ones who walk the aisles after the work is finished. You can imagine them as night gardeners moving through rows of delicate plants, checking leaves, tying supports, trimming damage. They do not hurry. They do not announce themselves. They simply maintain. Your cells are full of these caretakers. DNA is precious, but it is not fragile glass. It is durable, yet constantly exposed to motion, chemistry, and energy. Normal metabolism produces reactive molecules. Sunlight carries ultraviolet radiation. Even natural thermal vibration can nudge chemical bonds. So life does not rely on prevention alone. It invests deeply in repair. You feel this as reassurance, not that damage never happens, but that response is always present. You walk beside a strand of DNA and notice a tiny irregularity, a mismatched pair where the geometry bulges almost invisibly. The spiral is not quite smooth at this point. This is enough. The caretakers are sensitive to shape more than meaning. Distortion is their signal. A repair complex arrives, a cluster of proteins that recognizes the mismatch like a fingertip noticing a wrinkle in paper. They do not argue with it. They simply begin a gentle correction ritual. First, they identify which side is the newer copy, the one more likely to hold the error. Chemical markers and timing cues help them decide. Then a small section of the new strand is clipped out, not violently, but precisely, like removing a flawed stitch. The gap is reopened. A polymerase returns, this time not to copy the whole manuscript, but to patch a small place. Using the original strand as guide, it fills in the correct letters. A ligase enzyme, a molecular seamster, seals the backbone closed. Smooth again. Readable again. Stable again. Smooth again. Readable again. Stable again. This is mismatch repair, though you can think of it as post-copy editing. Quiet quality control after transcription by candlelight. You continue walking and encounter another kind of damage, not a mismatch, but a chemical alteration to a single base. Perhaps a reactive molecule has bumped into it and changed its structure slightly. The letter is still there, but its edges are wrong. Its pairing behavior is compromised. There is a different repair pathway for this base excision repair. A specialist enzyme patrols the strand looking for these altered shapes. When it finds one, it removes only the damaged base, not the whole step, just the letter itself, leaving the backbone temporarily empty at that point, like a missing tile in a mosaic. Other enzymes arrive to clean the site, insert the correct base, and reseal the structure. Minimal removal. Minimal disturbance. Elegant precision. You feel the tenderness of this, fix only what is broken, leave the rest untouched. Farther along, you sense a more noticeable injury, a bulky distortion where adjacent letters have fused together after ultraviolet light exposure. Two neighboring bases are stuck like melted wax. The strand bends slightly there. Reading machinery would stumble. This calls for a broader response: nucleotide excision repair. A team recognizes the distortion and removes a short stretch of the strand around it, a small patch, like cutting out a damaged square of fabric. Then, using the opposite strand as template, the gap is refilled and resealed. Cut. Copy. Patch. Smooth. The process is neither rushed nor emotional. Just practiced. You realize something soothing: repair is not rare emergency work. It is continuous maintenance. Thousands of small fixes happen in your cells every day without your awareness. Tiny corrections that preserve clarity. Silent rescues that keep the text legible. You are being edited gently, constantly, compassionately from within. You pause and notice that not all repair waits for damage to appear. Some systems write along with active processes. During DNA replication, proofreading happens immediately. During transcription, when RNA copies are made, surveillance systems monitor errors and stalled machinery. Maintenance is layered during, after, and between events. There are also guardians at a higher level, checkpoint systems that monitor overall DNA integrity before a cell is allowed to divide. You can imagine them as gatekeepers at a doorway between one room becoming two. If damage is too extensive, the gate does not open right away. Time is granted for repair. Division is delayed. Patience is built into the cycle. If repair succeeds, the cycle continues. If damage is beyond repair, the cell may choose a final path, a controlled self-retirement called apoptosis. Programmed cell death. Not collapse, choreography. The cell dismantles itself in an orderly way so neighboring tissue remains undisturbed. Even endings are structured. You do not feel fear here. Only respect. Because apoptosis is not punishment, it is protection at the community level. A way to prevent corrupted instructions from spreading. A librarian quietly removing a book that can no longer be restored. Most of the time, repair works beautifully. The vast majority of small DNA injuries are corrected before they ever influence protein building. Your genome is not a brittle relic, it is an actively maintained document. And yet you remember the earlier truth: repair is powerful, but not perfect. Sometimes damage escapes detection. Sometimes replication occurs before repair is complete. Sometimes the distortion is subtle enough to pass as acceptable geometry. And sometimes the repair machinery itself is temporarily overwhelmed by high damage load, by aging, by environmental stress. Guardians, too, have limits. You sit with this gently, not as vulnerability, but as realism. Living systems operate with extraordinary reliability, not absolute certainty. They are resilient through redundancy, not invulnerability. You watch the caretakers continue their rounds, scanning, clipping, filling, sealing, and feel a deep biological comfort. You are not left alone with your errors. You are not abandoned to molecular chance. You are supported by layers of attentive process you never have to consciously command. A self-healing manuscript. And still, in the quiet arithmetic of billions of letters and countless replications, a few edits remain, not corrected, not erased, small variations that slip past even the most devoted proofreaders, and begin a slower journey outward through time. You rest again beside the living manuscript, but now the walls of the library begin to whiten. The shelves stretch outward beyond your body, beyond a single lifetime, beyond even your species. The archive becomes a river, slow, continuous, flowing across generations. Until now, you have watched mutation at the scale of moments, one copy, one letter, one repair, one escape. Tonight, you watch what happens when tiny changes are allowed to travel through time. You imagine a single cell dividing, then its daughters dividing, then their daughters. A lineage unfolding like branches from a tree. Each division copies the manuscript. Each copy is nearly the same. Nearly, but not perfectly. A few letters differ here and there, like faint pencil marks in the margins. Most of these marks change nothing important. They ride along silently, generation after generation, like pebbles at the bottom of a stream. But time is patient, and streams become rivers. You float above this river of inheritance and begin to see that mutation is not only a risk, it is also a source. A source of difference. A source of variety. A source of new patterns for nature to test against reality. Without mutation, every copy would be identical forever. Perfect sameness. Perfect stillness. No variation to select from. No alternative designs to explore. Life would preserve, but it would not adapt. Change is the price of possibility. You watch populations now instead of single cells. Many individuals, each carrying slightly different versions of the same genes. Tiny spelling differences across the same instructions. Most differences are neutral, they neither help nor harm in the current environment. They simply exist. Variation is abundance at the microscopic level. You notice how environment acts like a gentle filter rather than a judge. Conditions shift temperature, food sources, pathogens, altitude, chemistry. Traits that were once neutral can become useful. Traits that were once useful can become less so. The meaning of a mutation depends on context, like a tool whose value depends on the task. A thicker fur gene matters in cold places. A salt-handling gene matters near oceans. An oxygen efficiency gene matters at high altitude. The letter change itself is small, but the environmental conversation around it gives it weight or quiet. You feel how relational biology is. Nothing acts alone. You drift through deep time, thousands, then millions of years, and see mutations layering like translucent paint washes. No single change transforms a species overnight. Instead, small differences accumulate, spread, fade, or stabilize depending on how they interact with survival and reproduction. Evolution is not a leap. It is a slow editing process. You picture a manuscript copied across centuries by many scribes. Each adds tiny variations, a phrasing here, a spelling there, a clarified line, a local expression. After enough generations, dialects emerge. After enough distance, new languages form. Not by sudden invention, but by gradual drift guided by use. Genomes drift too, but with selection acting as editor-in-chief. Variants that support survival tend to persist more often. Variants that strongly disrupt survival tend to vanish more often. Not perfectly, not instantly, but statistically, over time. Selection is gentle arithmetic, not dramatic decree. You sit with a comforting realization, Mutation is not trying to improve life. It is simply introducing variation. Improvement or disadvantage is assigned later by circumstance. Mutation writes options. Environment reads outcomes. Options first. Meaning later. You watch a mutation that slightly changes a receptor protein, just enough to make it harder for a certain virus to attach. In an environment where that virus is common, individuals with this tiny change are more likely to stay healthy long enough to reproduce. The variant spreads gradually through the population. A door opened by a letter change. Elsewhere, you see a mutation that allows an enzyme to function efficiently with a new food source. A new plant compound. A new sugar. Those who carry it gain nourishment where others cannot. The variant grows more frequent not because it is universally better, but because it is locally helpful. Advantage is always contextual. You also see mutations that are helpful in one setting but costly in another. A trait that conserves salt is useful in scarcity, harmful in abundance. A highly reactive immune setting protects against parasites but increases inflammatory illness risk when parasites are absent. No mutation is absolutely good or bad across all worlds. Only fitted or unfitted to the present one. You breathe slowly and feel the humility in that. Biology does not chase perfection, it negotiates with conditions. You notice something else: many evolutionary changes involve not new genes, but new regulation. Mutations and switches rather than tools. Changes in when, where, and how strongly genes are expressed. Timing adjustments. Volume adjustments. Developmental choreography rewritten by subtle promoter edits. Same instruments, different music. A gene active longer during growth changes limb proportion. A gene silenced earlier changes pigment pattern. Regulatory mutations are like lighting cues in a theater, they change the scene without changing the actors. The river flows on. You pass through eras where mass extinctions narrow diversity sharply, environmental upheavals acting like sudden bottlenecks. Only a subset of variants passes through. Afterward, mutation and variation begin expanding again, filling ecological space with new combinations. Contraction and expansion. Loss and invention. Always gradual at the letter level. You realize that every genome alive today is a palimpsest, a manuscript written, copied, lightly edited, and recopied across an unbroken chain stretching back to the earliest cells. No original draft survives, only descendants of descendants, each carrying traces of ancient mutations layered beneath recent ones. You are not just an individual text. You are a historical edition. Inside you are mutations that arose thousands of generations ago, now so common they are considered standard spelling. What once was variation has become baseline. Difference can become norm given enough time and spread. Today's mutation can be tomorrow's consensus. You rest in that continuity and feel mutation soften in meaning. Not merely error. Not merely risk. Also exploration. Also memory of adaptation. Also raw material for diversity, the reason no two genomes are exactly alike, even among siblings. Life writes in families of similarity, not clones of identity. The river widens further, and you begin to sense that while many mutations travel harmlessly through time, Some also travel into vulnerability, into conditions where altered proteins or regulation disturb balance within a body, and you feel the current gently bending toward how change, in certain places, can become illness, not as blame, but as biology unfolding. You continue floating along the long river of genetic change, and the water grows quieter here, not darker, not threatening, just more delicate. This is the place where variation sometimes meets vulnerability, where a small molecular edit, harmless in one context, becomes disruptive in another. You arrive gently. There is no alarm in this chapter, only understanding. Inside your cells, everything depends on cooperation. Proteins must fold correctly. Signals must arrive on time. Switches must turn on and off with rhythm. Growth must listen to boundaries. Repair must keep pace with wear. Health, at the molecular level, is not perfection, it is coordination. Illness often begins as miscoordination. You sit beside a newly made protein chain as it emerges from its cellular workshop. It is a long string of amino acids, like a ribbon with different textures along its length. Almost immediately, it begins to fold, not randomly, but guided by chemistry. Some segments attract water and face outward. Others avoid water and tuck inside. Charges attract and repel. Bonds form. Angles settle. Folding is function. But tonight, one amino acid in the chain is different, replaced because a codon changed through mutation. Just one piece swapped. Sometimes this makes no difference at all. But sometimes, depending on where it sits, the fold shifts. A pocket becomes shallow. A hinge becomes stiff. A docking surface becomes rough. The protein still exists, but it behaves differently. You imagine a key cut slightly off at one tooth. It may still enter the lock but turn poorly. Or only sometimes. Or only under pressure. In biology, partial function is common, not just on and off, but gradients of effectiveness. Many genetic diseases are not absolute failures, they are efficiency changes. You watch an enzyme that normally converts one molecule into another as part of a metabolic pathway. Because of a mutation, its active site, the working pocket, binds less tightly. The reaction still occurs, but more slowly. Upstream molecules accumulate. Downstream products run low. The pathway becomes imbalanced. Chemistry feels the slowdown. Cells respond with feedback loops, increasing production, activating alternate routes, adjusting demand. Sometimes compensation works beautifully. Sometimes it is not enough in certain tissues or conditions. Symptoms appear not because the system stopped, but because it strained. Strain is often the biology of illness. You walk onward and observe structural proteins, the fibers that give cells and tissues shape and resilience. Collagen cables. Cytoskeletal scaffolds. Membrane anchors. A mutation alters how one component links to another. The structure holds, but with reduced tensile strength. Over time, tissues that experience repeated stress show wear more quickly. Not collapse, but fragility. You notice something important: the same mutation can have different effects in different cell types. A protein critical in muscle may be less critical in skin. A receptor vital in neurons may be optional in liver cells. Expression patterns matter. Location matters. Context always matters. You rest beside a signaling pathway, a relay of proteins passing messages from a receptor at the cell surface to the nucleus inside. Each step activates the next like falling dominoes. A mutation causes one relay protein to remain slightly active even when no signal arrives. The message whispers when it should be silent. Background noise enters the channel. Cells interpret persistent low-level signals differently from clean pulses. Gene expression shifts. Behavior changes. Growth decisions adjust. Over long periods, this kind of misregulation can contribute to disease states not as invasion, but as miscommunication. Illness as crossed wires. You feel how helpful it is to describe this gently, not as betrayal by the body, but as altered messaging inside a vast network. The system is still trying to function, still trying to balance, still responding with the tools it has. Some mutations influence how cells control division. Normally, growth is governed by checkpoints, feedback signals, and neighbor awareness. Divide when needed. Pause when crowded. Repair before copying. A mutation in a growth control gene can loosen one of these brakes or press too firmly on an accelerator. Growth loses rhythm. Even here, progression is rarely immediate. Multiple regulatory layers must be bypassed before uncontrolled growth persists. Cancer, for example, is not usually born from a single mutation, but from an accumulation, several edits across time affecting growth, repair, and survival pathways together. Layered change, layered consequence. You breathe slowly and notice how often biology builds backups. Two copies of many genes. Parallel pathways. Redundant controls. This is why many harmful mutations remain recessive, their effects hidden when a working copy is present. Only when both copies carry the disruptive change does the full effect appear. Redundancy is mercy. You also observe mutations in genes involved in DNA repair itself. When proofreading systems are weakened, mutation rates can rise, not explosively, but measurably. The manuscript gains edits faster than usual. Variation increases. Risk increases. Not destiny, but probability. Even guardians need guardians. You pause and let compassion fill the space. Genetic illness is not punishment, not intention, not moral narrative. It is molecular outcome, chemistry meeting context across time. Understanding removes blame and replaces it with clarity. And clarity is calming. You notice, too, that many mutation-linked conditions today are treatable or manageable because science has learned to read the manuscript. Therapies can replace missing products, adjust pathways, support folding, modulate signals. Knowledge becomes intervention. Intervention becomes relief. Reading leads to response. You sit with a final gentle truth. Mutation can lead to illness, yes, but it can also lead to resilience, protection, and adaptation, as you have already seen. The same mechanism produces both vulnerability and strength depending on placement and environment. Variation is neutral. Outcome is relational. As the river carries you forward, your fear dissolves into understanding. Change is not the enemy. Unawareness is. You drift further along the river of inheritance, leaving the delicate ripples of illness behind, and notice new light playing across the water. It is quiet, soft, yet unmistakably vibrant, the glow of beneficial change. Here, mutations do not disrupt, they enhance. They whisper possibility instead of warning. They are the friendly edits, the hidden gifts, the subtle experiments that life tries quietly, generation after generation. You see, mutation is blind. It does not intend to help or harm. It simply introduces variation. But in certain contexts, some variations turn into advantages, tiny strokes that give organisms an edge, sometimes in ways that only appear over time. You float beside a population of cells and imagine a single letter change producing a slightly altered receptor protein on a cell surface. This tiny adjustment makes it more difficult for a certain pathogen to attach. The cells survive longer, replicate more efficiently, and the variation spreads. No heroism is needed. Chemistry and chance have simply favored a particular outcome. You notice another case, a protein in the liver that processes a plant toxin more effectively because of a one-letter change. In environments rich with that plant, the individual carrying this variant thrives, quietly outcompeting those without it. Again, nothing dramatic. No rush. Just subtle survival. Adaptation is poetry in motion. You realize these friendly mutations are rare compared to the silent or neutral ones. Most variations drift unnoticed, quietly riding along in genomes like unnoticed punctuation. But the few that interact positively with the environment become whispers of resilience. They accumulate, layer upon layer, across generations, contributing to population diversity. You imagine a child born in high-altitude mountains. One amino acid change allows hemoglobin to carry oxygen more efficiently. Life flows more easily through each breath. That single molecular adjustment matters little in the valley, but here, it matters profoundly. One small letter change can meet a vast challenge. You sense that biology is always experimenting. Every replication, every division, every offspring carries the potential for novelty. Some novelty fades. Some survives. Some thrives. Life is both conservative and exploratory, conserving what works, testing what might work better. Mutation is the brushstroke, selection is the eye that decides which strokes stay. You wander through these examples like walking through an art gallery of molecular chance. Sickle cell trait in areas with malaria, a mutation that confers protection at a cost. Lactase persistence, a single regulatory change allowing adults to digest milk, emerging independently in multiple human populations. Pigmentation variations, small genetic edits that provide camouflage or UV protection in different environments. You notice that even minor changes can produce major ripple effects. One amino acid swap in a protein may subtly alter the immune response, making a population more resilient to local infections. One regulatory change may tweak the timing of developmental genes, influencing growth or behavior in ways that confer advantage. Life constantly explores these small variations. And yet, these benefits are context-dependent. A mutation that is protective in one environment might be neutral or even detrimental in another. Biology does not assign universal value, it only tests compatibility with circumstance. You feel comfort in this impartiality. Nature is curious, not judgmental. You see how populations carry a mosaic of these friendly edits, quietly balanced alongside neutral and occasionally harmful mutations. diversity becomes a reservoir. Hidden advantages can emerge when conditions shift. Chance gifts may lie dormant for generations, ready to flourish when the world changes. Mutation is patient. Life is patient. You rest beside a glowing filament of DNA and imagine all the potential tucked in there, subtle differences waiting, quiet variations that may help tomorrow. You feel gratitude for this hidden generosity. Mutation is not only a source of risk. It is also a source of resilience, ingenuity, and creativity written in molecular ink. You sense a soft rhythm emerging from the patterns, a pulse of possibility. Each friendly mutation, each hidden gift, is a note in a symphony of adaptation. Some notes harmonize immediately, some lie dormant, some resonate only under the right conditions. The melody is ongoing, improvisational, patient. And as the light of these beneficial changes warms your awareness, you feel your trust in variation deepen. Mutation is not merely error or misstep. It is nature's quiet hand exploring what might be possible, a canvas for adaptation, a whisper of resilience in the ever-moving flow of life. You close your eyes gently and sense that some of the most profound gifts are yet to be discovered, tiny molecular edits waiting to be revealed in the next chapter, where we learn how science begins to read and even touch this intimate language of life. You now leave the quiet riverbanks of natural mutation and drift toward a different space, a place where human curiosity enters the molecular manuscript. Here, the library of life is no longer only observed from afar. Scientists have learned not just to read it, but also to annotate, interpret, and, in some cases, even edit it. The first thing you notice is the act of reading. DNA sequencing is like listening to the whispers of the library at the smallest scale. Imagine hundreds of millions of tiny letters, each encoded in a chemical language, being read aloud simultaneously. The sequence emerges not as a blur but as a melody, a text, a series of patterns waiting to be understood. You feel the rhythm of it, each letter, each codon, a soft note in a grand composition. Sequencing is not fast in the human sense. It is careful, meticulous, accurate. Machines and enzymes work together to transform chemical signals into readable letters A, T, C, G, unfolding the story of an organism in intricate detail. Reading is the first act of understanding. Only by listening carefully can errors, variations, and hidden messages be discerned. Scientists can compare sequences across individuals, populations, and even species. They can map the subtle changes that produce disease, the friendly mutations that confer advantage, and the silent letters that hide in margins for generations. You sense the wonder in this act, the intimacy of listening to billions of years of evolutionary experimentation compressed into the present. But reading is not the only thing possible. Editing enters softly, like a feather placed on a page rather than a hammer striking a wall. The advent of tools like CRISPR-Cas systems allows targeted edits in the genetic manuscript. Scientists can now locate a specific letter or a small group of letters and replace, remove, or adjust them with unprecedented precision. You imagine this as a molecular calligraphy brush guided by both logic and care. A single triplet can be swapped. A regulatory sequence nudged. A problematic insertion excised. The manuscript responds because it has always been chemical, malleable, yet guided by rules. The responsibility of this power is immense. You feel it as a quiet weight, the knowledge that editing a genome is not merely writing a word differently, but potentially influencing life's course in subtle or profound ways. Precision is essential. Understanding is vital. The ethical dimension hums beneath every molecular adjustment. You drift along watching repair-like enzymes being guided intentionally, as if the cell itself were cooperating with human instruction. Nature's caretakers, the proofreaders and polymerases you met earlier, continue to monitor. Editing is integrated, not chaotic. The system is still alive, still responding, still resilient. There is awe in this process. The ability to read and interpret the genetic manuscript transforms curiosity into capability. Knowledge becomes agency. And yet, even in the presence of such capability, humility is required. The manuscript is vast, intricate, and its consequences are not always predictable. You notice a gentle rhythm, the interplay of observation, understanding, and careful intervention. Each read brings insight. Each edit requires verification. Each result informs the next step. The process is deliberate, iterative, and respectful of complexity. You imagine watching a disease-causing mutation being located, then replaced in a single cell. The protein it encodes begins folding normally again. Signals transmit correctly. The cellular pathway resumes its measured rhythm. Intervention is possible, and yet it remains soft, cautious, experimental. And it is not just disease. Scientists can explore adaptation, variation, and resilience. Friendly mutations, silent letters, regulatory edits all can be examined, understood, and, in theory, guided. You breathe slowly, feeling both humility and wonder. The manuscript of life is no longer untouchable. Observation has become dialogue. Reading has become listening. Editing has become conversation, careful, precise, and weighted with responsibility. You sense the connection between knowing and caring. Science does not dominate the code. It partners with it, learning its rhythms, anticipating its reactions, respecting its resilience. It is a collaboration across scales: molecular, cellular, organismal, and ethical. And as you drift away from the microscales and feel the river widen into possibilities, you begin to notice that every change, whether natural or guided, leaves traces subtle, persistent, sometimes unpredictable. The genome carries its history forward, integrating human edits into the ongoing flow of life. And in that realization, you feel a quiet awe that life is not merely observed anymore. It is touched, understood, and occasionally guided, but always with reverence, like turning pages in a library where each word carries both inheritance and potential. Some edits bring healing, some bring insight. And beyond these, countless mutations still drift quietly through the manuscript, waiting to be discovered, understood, and perhaps gently nudged in the next chapter of life's unfolding story. You linger in the library of life, surrounded now by both the massive tomes of genomes and the delicate, almost invisible notes scribbled along the margins. These are the living annotations, the edits, mutations, and variations that do not always command attention, but subtly influence the meaning of the text. They are marginalia in the manuscript of existence. You walk softly along a DNA strand and notice a single letter change from centuries past. It has traveled through countless cell divisions, through generations, carried silently by inheritance. To the untrained eye, it is barely noticeable. To the patient observer, it is a note, a tiny suggestion, a possibility, a record of exploration. Some margin notes are purely neutral scribbles left by chance. They do not alter the function of the protein, the message of a regulatory sequence, or the rhythm of development. They are whispers of history, footprints of replication, quietly enriching the manuscript without demanding consequence. Other notes are more meaningful. They guide, influence, and occasionally redirect. A slight amino acid change alters the way a protein folds, subtly improving stability or activity. A regulatory mutation adjusts timing, ensuring expression occurs at the right moment. These are intentional only in the sense that life experiments, they are echoes of nature's gentle hand exploring possibility. You feel the dynamic nature of this document. No genome is static. DNA is not a fixed, inert code, but a living manuscript, responsive, adaptive, and evolving. Mutations accumulate, interact, and layer upon one another. Each organism carries its own unique collection of margin notes, some inherited, some new, some fleeting, and some persistent. You consider your own body in this light. Every cell carries its own manuscript, slightly varied, gently edited by time and environment. You are a living archive, a compilation of letters and notes, a dynamic story still being written. The manuscript is yours, yet connected to countless others across generations. You notice that margin notes can sometimes interact in ways unexpected. Two neutral changes may together create a beneficial effect. A subtle edit that seemed harmless in one context may become protective in another. Interactions are complex, patterns emerge slowly, and the manuscript gains depth beyond simple linear reading. Even harmful changes can be illuminating. They reveal the fragility of certain pathways, the critical points of folding and regulation, the places where biology must remain vigilant. They inform understanding, guide research, and help shape interventions. Notes at the margins are rarely wasted. They are lessons, even when they signal caution. You drift gently through this landscape and realize that life itself is a dialogue between stability and change. The main text preserves essential function. The margin notes provide creativity, exploration, and adaptability. Together, they create a manuscript capable of resilience, growth, and evolution. You pause at a stretch of DNA rich with variation, a forest of edits stretching along the helix. Each note, each mutation, tells a story. Some remain silent. Some shape development. Some persist through generations. They are subtle testaments to the patience of life, the slow accumulation of experience, the quiet power of adaptation. You feel a deep sense of connection. Not only are these notes part of biology's history, they are part of you. Every cell, every gene, every subtle variation contributes to the story you carry, the functions you perform, and the adaptations you express. You are a living, breathing manuscript, still evolving, still edited, still alive. And yet, the margins are not merely historical. They are forward-looking. Each new mutation, each new edit, is a potential story for tomorrow. Some may change nothing. Some may quietly reshape function. Some may ripple across time to influence future generations. Life is a manuscript that grows, learns, and explores through the gentle accumulation of these margin notes. You rest with the thought that your own being is part of this dynamic story, a living text still being revised, still being interpreted, still being shaped by both chance and context. The writing is ongoing. The pages are not complete. And somewhere beyond the visible letters and familiar edits, new notes are forming, quietly, patiently, ready to shape the next chapter of the ever-unfolding story. And as you close your eyes, you sense the soft anticipation of those future edits, the next whispers in the margins, waiting to ripple outward through time and life. You float gently at the confluence of everything you have explored: the manuscript of DNA, the river of mutation, the guardians of repair, and the quiet experiments of friendly variation. Here, the threads of all chapters intertwine. You feel the power of a single change, a single letter in the vast text, and how it can ripple outward like a pebble dropped into a still pond. Imagine a single molecular letter swapping in one cell. It seems insignificant at first, almost invisible, like a whispered secret in a library hall. But the cell divides. The change is copied. Daughter cells carry it. Some remain local, some migrate. Tissues subtly adapt to the new signal, the new protein, the new rhythm. The effect spreads, quietly but steadily. Now imagine this change occurring in a reproductive cell, an egg or sperm. Suddenly, the ripple extends to an entirely new organism. The letter that shifted in one microscopic place becomes part of a whole new body, a whole new life. In that child, the edit interacts with countless other letters, pathways, and environmental contexts, producing outcomes both predictable and mysterious. Time stretches outward. Generations fold over generations. That single change can persist, fade, or amplify depending on circumstance, environment, and the interplay of other variations. One tiny alteration may never be noticed, or it may subtly influence survival, reproduction, and adaptation across decades, centuries, or millennia. You feel a gentle awe, the interconnectedness of all life, the delicate balance between chance and consequence. Nothing exists in isolation. Molecules, cells, organisms, populations, ecosystems, each participates in a conversation written over eons. Each small mutation contributes to the collective story, like a note in an infinite symphony. Some changes ripple outward to influence health, form, or function in a body. Others ripple through populations, shaping diversity, resilience, and the potential for future adaptation. Every living organism carries the echoes of countless past edits, and every organism contributes to the possibility of future ones. Life is a web of letters, a network of edits, a living document stretching across time and space. You feel gratitude for this resilience, this layered complexity. Life has built redundancy, repair, and adaptability into the very fabric of its manuscript. Variations, edits, errors, and corrections coexist, each playing a role in the ongoing story. Even the smallest change matters, because its ripples can reach farther than you can see. And in this awareness, you sense your own participation. You are not separate from the manuscript. You are one of the many living pages, carrying edits from past generations, contributing your own subtle variations, and passing them forward. The poem of life continues through you, just as it continues through every cell and organism around you. The river of time flows endlessly. Mutations arise, some fade, some persist, some transform. Friendly, harmful, silent, profound, they are all notes in the manuscript, each capable of changing the course of life in ways both gentle and immense. The story is never static. The writing continues, patient and persistent, across every scale, from molecule to body to lineage. And as your thoughts soften, drifting toward the rhythm of sleep, you rest in the quiet understanding that a single letter, tiny, almost imperceptible, can indeed shape not only a life but a lineage, carrying possibility, resilience, and wonder across generations. Each change is a ripple, each ripple a story, and each story flows onward, unending, into the vastness of living time. In this gentle truth, you feel the calm satisfaction of connection, and you drift, knowing that the manuscript, like you, is always still being written,